Good afternoon to everyone. I welcome all of you to the 94th lecture in the lecture series in Nonlinear Dynamics conducted by the Department of Nonlinear Dynamics, Bharathidasan University, with the support from TUSA 2.0. It is my pleasure to introduce today's speaker, Dr. Samrat Chatterjee. Dr. Chatterjee received his doctorate from Jodhpur University in the year 2008. He completed his postdoc in the following year in the University of Torino, Italy. I think it is one of the beautiful cities in Italy. He then moved as a research scientist in International Center for Genetic Engineering and Biotechnology, New Delhi, for the period 2009 to 2012. He started his teaching career in 2013 as an assistant professor in Translational Health Science and Technology Institute, and he is serving as an associate professor in the same institute now. He is a life member of Biomathematical Society of India and Indian Science Congress Association. He is also a member of International Society for Computational Biology. His autobiography is published in 2000 edition of Who's Who in the World. He has several notable publications in his area of expertise. He has given several lectures and talks in many conferences across the world. He has authored 70 publications in peer-reviewed journals and 8 book chapters in his name. With this short introduction, now I invite Dr. Chatterjee to deliver his lecture. Over to you, sir.
Thank you, sir. Thank you for this nice introduction. And it's not a short, it's a long one. So not done much. But if I understand, today's lecture is mainly aiming for the students.
Yes, exactly.
That they can get encouraged and take this field or something.
Yes, exactly.
So I have prepared my slide like that. So I will start with some basic things so that they will know what kind of area mathematics they can use. And then I will take some of the work which I have done recently as an example to where they can actually apply this field. So I think they are all from mathematics background or physics background?
Physics.
Physics background. Oh, okay. So maybe I start my presentation?
Yes.
So sorry. It is visible?
Yeah.
Okay.
Yes.
Okay.
Nice. Yes.
So actually, THST where I'm working at present is a Translation Health Science and Technology Institute. So they mainly work on different diseases and therapeutic. They try to understand the therapeutic what will be the targets, what will be the drugs, and all those things. So this is a very good area where we mathematicians or physicists can actually help them, and that is one of the motivation of today's talk to the students here. That my background, as sir already described, is from mathematics. So I did everything from pure mathematics background till my postdoc, and then I shifted to this biological field. And today's biology, you can think of like 100 years back of physics, because a lot of open questions are there and a lot of complicated mathematical and computation tools you can apply, and those things are there. So I choose today for the presentation the type 2 diabetes biology. I will just try to tell you what kind of, I will not only tell you that what we have done in terms of mathematics or computational, but actually what in real field it means, because some of these results have already experimentally validated and they have been taken forward. So this is why I choose this field. And two major areas I will just go very briefly. I will not go in details of them, but what I want the students to know the keywords in today's lectures. If they can write it down somewhere, and when they will actually pick up the field, they will know it. So two major keyword in this title, you will find the mathematical modeling and network analysis. So basically these are two very different fields, but I will try to show you what kind of applications they can be done in real biological problems and what kind of analysis we can get from there. So if I start with my research experience, it's very confusing because frankly speaking, my biomathematics journey has already started when I joined ISI in 2003. But that was mainly, if I be realistic, it's mainly mathematical modeling and mathematical tools. We take the ecological problems, but those are not I will not say that we are more focused on the mathematical part. But then when I moved to Torino, as I said, it's really a beautiful city. It's in the Alps. And there I learned a lot on the computation simulation part and all those things. And then when I came back to India, fortunately, I got a job where I can apply this mathematical modeling and computer simulation in real biological problems. So fortunately or unfortunately, three different experiences have gave me this kind of a confusing biomathematician standard. So one can take this one, and it's vice versa, because sometimes I also give lecture to the biologist background students, and I have the experience of working with them. So even I have one of my students who is from Italy, and he is actually a biologist, but then he actually make mathematical models, solved them, and did publish paper for his PhD. And of course, for the mathematician, we always have that edge, because if you know the mathematical part, you just need to understand the biology. You can talk to, and you need to understand that much of biology which you can actually talk to them, you can actually convey, because they are from very different domains. So you need to understand their language, and they need to understand your language. So this is only the challenging part, which I will try to show you through this presentation, that what kind of problems or what kind of language between these two domains actually talk with each other. So let's start with some basic mathematics, because I think this is just to show you that these are the things which you are already doing. I mean, even if you're from physics background or this kind of analysis or this domain of mathematics you have seen. So I will just take you from here, and I will gradually take you towards the biology part. So let's start with so the very first thing is, of course, what is dynamical system. So to understand dynamical system, we need to know the differential equations. And I think since we have already 90 lectures before me, so everybody must have given you this kind of background. But just bear with me, and I will very briefly take you there, but then you will actually understand that whatever you are doing today, whatever the analysis you are doing, it may seems very theoretical and it may seem far from your biology, but I will try to take you through this journey, that how this kind of theoretical stuff can actually brings real field applications and can make the biologists also understand what you are doing. So let's start with this dynamical system. So we all know the autonomous differential equation. It will have, and this is, of course, we are talking about the first degree differential equation. So let's just go very slow, very fast. These things. This is a linear system. I'm just making R square, two dimension. So you have x1 dot, x2 dot. They have this x1 plus bx2 and cx1 dx2. This is typically a linear system in R2. You can make it a matrix form. And the thing what we started actually is to solve these equations. So we know that, I mean, normally from class after class 10, from 11, 12, when we start doing calculus, we start looking at the solutions of differential, first differentiation and then differential equations. But now when it get little complex, we start to know when you actually can't solve it, then you start to know what it could be the nature of the solution. So the things which next we do in these kind of things, assuming the system would be complex, we go for the qualitative analysis of the system. That if you can't find exact solution of a system, but at least you will know that what kind of solution or the nature of the solution of your system. And for that, couple of things which you do, and these are well known to you people also. Equilibrium point we try to find any system, and that is the point when your dx dt becomes 0, that side, and then it becomes an algebraic equation. You solve it, and then you go for the stability of the equilibrium point. Now this is very important, because equilibrium, you may find a lot of equilibrium points in your system, but mainly the attractor, which is called in mathematical things, or which is sink you can call, or stable point, they are the point where ultimately your solution goes. The other points actually reflect it back, but that is the, so normally we then look for the nature of the equilibrium point. We find we take the Jacobian matrix, we find the eigenvalues. Of course, you know the real part of the eigenvalues, depending on the nature of the real part. If it is negative or positive, it gives the equilibrium point, and so on. So this, I think you all know. For example, if you have a stable focus, your real part of the equilibrium point, if it is less than a negative, it will be a stable focus. You look like this. If they become zero, I mean, if you have a really if you have purely imaginary roots, it will become center, it will start oscillating. So these are the solution which you find for a differential equation when you can't solve it. Now, just this is one example I normally use, because when we talk about these things, we need to understand actually that what is this equilibrium point and what is actually stable and unstable equilibrium point. I mean, those things can be mathematically well defined, but can we see it? We can see it in a lot of places, but an easy up is the pendulum, for example. So if you see this pendulum, so this pendulum actually have 3 points, if you see. So A, B, and C. B is here. So since I'm not there physically, because I would have asked this question to the students, but so the thing is that here normally I ask the student that how many equilibrium points are there, and sometimes they say 3, sometimes 2, sometimes 1. So but the thing is that here if you see, sorry, here if you see the equilibrium point, this is actually 3 equilibrium points are there. So by definition, A, B, C all are equilibrium points, because if you try to imagine that at that point, at that very minute, the dynamics of this equilibrium point stops. But then the problem is that nothing stays at A and C. B, you can imagine that, okay, after a certain time period, the pendulum will stop in B, and so it is an equilibrium point. But why A and C? Because A and C is the point where, if you can imagine, there this X dot become actually 0. But it is not a stable equilibrium point, which is different between B and A and C. So from A and C, it will again come back, while from B, it will stay there. So for example, if you take a cup, I think you all can see me. So if you take a cup and put a marble here in the bottom of the cup, and if you try to take that marble along the surface of the cup to the top portion and leave it, it will come back to that portion. But if you just put the cut like this and take a marble at the top and put this slightly shift the marble from here, it will never come back. So that is how you can actually think of a stable and unstable equilibrium point. That one is actually attracting the thing, the other one, if perturbed, it will never come back. So knowing all the differential equation, there are other things also, bifurcation point and all those things, which you must have read in your things in your paper. So knowing all these things, now the questions come up. All these things are there. Then what is actually the use of mathematics in biology? Because this is at the end, your system, if you are really working in the biomathematics or mathematical biology, your system will be biology. Even if you call it a biophysics, or your system will be biology, and your tools will be your mathematics or physics. In this case, I'm talking about the mathematical tools. So what is the role of mathematics in biology? As I told you, you can think of biological problem today is like 100 years back in the physics problem. Everything is open up, there are a lot of things coming, and all those areas are coming. So all it's began with simple things, which I just wrote here, that we try to find out the change in most of the cases, the change in the population density, for example, suppose. So what is actually a differential equation? So differential equation is, crudely speaking, your one of your variable is changing, which depends on the other variable, and the other variable is independently changing. So in a dynamical system, you can think of time as the independent variable. I mean, it is the independent variable which is changing, and anything that changes over time, in this case the population, we draw it here. So suppose here, for example, if n is representing bacterial density, time is of course the function, then what actually we are trying to find out here, if you try to now know that where this mathematical concept is going to the biology, is that we are trying to find out the change in the bacterial population, for example, bacterial population over time. And this change which we are trying to find out here is a continuous change. Now this could be a little bit problem when you are actually doing the experiment, because there your changes always will be discrete, it will be at different time. But now we are making that whole system to a mathematical point where we are actually make it more realistic, and we are trying to find the change in that bacterial density over time. So this is the way you are now going towards the biology. So how it all begins? So there is, I think these models you have already heard about. So there's a Malthus model in 1798 by the English political economist Thomas Malthus, what he had tried to understand the US population, which grows for over like 100 years. So he made a mathematical formula, and he actually showed that mathematical formula is satisfying the data point of that population. So he simply wrote dpdt, change in population over time, is equal to r naught into p, where r naught is birth minus death rate plus in some cases migration. But the problem with this one, if you really go with this, that this model, if you solve, it doesn't have I mean, it will keep continuous going, it doesn't have any stable point, for example, if you say, or it is curving down. So in later a Belgium mathematician Perivos modified this one, and he just proposed or modified this one and proposed a logistic model. So here you can see this one. So in his case, what it did, the dpdt r naught p was same as the last model. He just put a minus term, minus p by k. So this minus p by k is actually taking two things together. One is this p and p showing the competition between them, and the other one is the k is the carrying capacity. So every system has, if I talk about an ecology or anywhere, every system has a carrying capacity, means it can, the maximum amount of population it can hold there. So for example, if you take a pond, it must have the maximum amount of fish it can hold. So if the fish population is less than that, it will grow up. But if the fish population is more than that, it will die. Or even if you don't fish it out, they will die out at some point. This is called the carrying capacity of any system. So he added that one, and then he of course proved this in growth of yeast cells. Then in 1929, 1930, there was two papers separately done by Lotka and Volta, who actually did it in a two species. So till now we are talking about a single species. They did a two species. So what it happened that they've seen that there is a data that shows that relation between a hare and a lynx is that if you take their populations over a period of almost like 100 years, their population shows a kind of oscillations. If you see this graph, so one is going up, the other going down, and so on. So to satisfy this, they then finally propose a model here. So this is a Lotka Volta, famous Lotka Volta model, later name on. They actually published in a different papers, but later it becomes. So they showed that this dx dt equal to x minus bxy, and dy dt is minus cy plus pxy. And if you solve this, because I told you, like, you find the equilibrium point, then you find the Jacobian matrix, you look at the eigenvalues and all, you will actually get that they are showing a oscillation. Actually, they show the center. So people have started using mathematical models and other things in biology from very beginning, but with but they are mainly focusing on the ecology or in the large scale populations. But with development of new machines in the biological field, which I will just show you, that a lot of informations are coming up in terms of the healthcare, in medicals, and so this now become more focused in the field of that clinical and healthcare. So before going to that, so let me tell you what I was telling you in the beginning, that what actually mathematical biology is. So mathematical biology. If you want to do that, first thing you need to talk to a biologist. You need to understand their problem. So once you start talking to a biologist, you understand their problem. Then you sit and make a model, mathematical model. That mathematical model is nothing but a mathematical language of the same thing what the biologist had told you. Now here a catch is that when you really start making model, you have to actually have a trade-off. Whether you really go for a complex model which will be more realistic, then that becomes a little difficult to solve. I mean, how much you will solve, how much information you will get, because I'm not talking about the computational but analytically. And the other way around, how much simple you will make it so that you can solve it, get all the information, but then it will become far from real. So that is a trade-off a mathematical modeler had to think, had to discuss with the biologist, make the assumptions which satisfy them also, and make your model simple, and so on. Then of course when you have the model, you go for the simulation, you go for comparing the result with the experimental validation. I will give you an example in my talk, and you'll find it. And then you, of course, make a new hypothesis, you suggest new experiment, and so on. So this is how actually mathematical models can fit in the biology. So this is if you can think it, this is like this. It starts with the experiment or data, then it goes to mathematical model, then you solve that model, make simulations, and then again go to experiments. And it is not like it's always start with the experiment or the data. That is why I make it a circle. It can start with your knowledge also. You can read something, you can made out of mathematical model, then you do all these things and go back to the biologist and tell them that this is what I have read, then this is what I did, and we got this. Can you check it out? So even a biologist can also start, and then you can help them, and in other way around, you can also start, and then you can take help of the biologist. Now, very briefly, I'm just telling you the other keyword which I have put in my title, the network analysis. So network analysis, actually, if you see, it is actually the graph theory in the mathematical subject, the graph theory and part of that domain. So in network analysis, if I start again, if I see his history, there is a very nice history of this network analysis. So it all began in like 200-300 years back, when there was a king in Prussia, which is like in Russia. So he proposed a problem. The problem was very unique. So the city was actually divided by the rivers, and there were actually 7 bridges that connect the part of the city over the river. So what he has asked that he proposed a problem. He told that suppose if I start from one point, what is the probability that I've crossed all the points and come back to that point again? And the condition is that all the points I will cross once. So he proposed that. Many people have tried. Late in 1736, Euler has actually solved that problem, and you can think that as the first graph network analysis what he has done. So what he proposed it, he put that problem in this form, where he put that bridge as the edges, and the point of the starting where these edges are connecting as the nodes. So here you can see that these are the points which the edges has started, and he termed this as the node and edges. Then what he has solved is that if this number of edges are even number, then the condition for if you start from one point, crossing all the edges and come back to that point, will only satisfy if the number of edges are even. So in that actual problem, since the number of edges are odd, so it doesn't have any solution. That means you will never get, if you start from one point, whatever the condition you put, you will never come back to that point, crossing all the bridges once. So this is a very interesting history I thought to share with you, where this network analysis started. Then of course this network analysis, the graph theory which I told you, it is the graph is defined by this V and E, which are the vertices and edges. And this is actually a lot of places where this graph theory has already started. In biology it came late. Earlier it already been used in air routes, in road networks, then of course in your computational, this your internet and all, these are all through this graph theory is maintained. But it is getting a focus in the biology, which is now coming as because of this development of new tools, which I have just told you. Because in last 20 or 30 years, there are a lot of big data are coming up because of the discovery of mass spec, and then you have these proteomics, you have metabolomics, you have RNA-seq data. So the biology, the way the people looking at the biology has changes a lot. Now it's become much more, I can say in the terms like much more mathematical or physics friendly. Because when you see this data, when you look at this, you will find that is another physical system, but much more complex. Whatever my experience is, that it's really very complex, and lot of things are, because the problem is that still a lot of things are unknown, which is actually also good for us, in the sense that a lot of problems you will find here. So as I told you, with this lot of biological data coming up, this network analysis is getting more focused on this field. And lot of biological lab, actually they themselves are trying to do this network analysis, because a lot of softwares are available at certain level, they do it. But of course, when you really go for advanced level, you need to know the mathematics behind those network analysis. So there actually you can play a role, because still now lot of, like in mathematical models, you can make a lot of mathematical models are available. Similarly here also, there is very less algorithm is known. There is a lot of algorithms you can actually build. When you actually look at the data, if you try to solve any problems, you can build your own algorithm based on network analysis, and you can go ahead. As I told you, for example, this protein protein interaction network. So here actually how two proteins interact with each other, because I just give you a little bit of biological background, because this is something might be new for you. So when actually in cells, when this protein works, or how the cell works is, when these proteins actually interact with each other. So when there is some signal is coming out from outside or within the cells, there is a very particular pathways, and all those paths are already mentioned. So a particular protein of this path get perturbed. Suppose some kind of chemical reactions happen there, then it actually go to another protein. If I say very crudely, in the sense that information is passed to another protein in a very, in a biochemical reaction, but you can think of that. And similarly, all these proteins actually follow a path. They sometimes this information goes to the nucleus, then they take the whatever decision the nucleus want to take, and then again goes back to the protein and take the action. So it's a very very good example of signaling network. So there are a lot of works have been done in cell signaling network. We also have an interest on that one, because there you can actually use stochastic network, you can use noise to understand those things. But anyway, that's not in today's lecture. But there are a lot of cell signaling network work you can do. So this is a place where this network analysis is coming. Now, giving all this background very briefly, let me just go through you some of the applications which we did during our work. So you'll see that all this theoretical stuff can actually, how they are helpful to a biologist. Not that we are doing something, it's really help them, because something which we can find as a mathematician or physicist is very difficult for a biologist to find it doing only experiment, because that is time taking, that is costly. Sometimes logistic reasons are main for doing that, and we can actually just sit there, use our head, and we can actually go through. So to start with, the first example which I show you. Okay, before going to that, maybe a couple of slides, I will just give you the importance of why I choose this topic, or why our lab is working on this. As I told you, there are 2 3 things on which we are working on. One is cell signaling, one is microbial tuberculosis. So diabetes is one of the very important I will say disease in today's world, in the sense that it is a slow killer and silent killer. In the sense, I will show you how it does it. And it is actually increasing all over the world. In fact, in India, people are saying that India is a diabetic capital of the world. So it has almost 17% of the total number of diabetic patients in the world. There are around currently close to 80 million people with diabetes in India, and of course this number is going to increase a lot in future. And the problem with the diabetes, that of course we know, in case of diabetes, that we have the insulin secretion reduces, causing the increase in the glucose. But there are of course different type of diabetes: type 1, type 2, gestation diabetes, pre-diabetes, and all. So today's my talk will be mainly on the type 2 diabetes, because the problem with this is that what happen actually, what does glucose does in our body? When it goes in our body, it goes from blood, it goes to a different cells, and using that glucose, different cells actually functions. Now problem in some cells, this entry of the glucose into the cell depends on the insulin. And in those cells, what happens, we call it insulin resistance. There kind of some problem arises in the insulin, I will say the path which will use to make the glucose inside the cell. And as a result, what happened, the glucose doesn't go inside the cell, it remains in the blood. That is why when we take the glucose from the blood, we find it more. And actually this start with insulin resistance, but then gradually what happened, it has created toxicity in the beta cell of pancreas, and that result in insulin resistance, insulin reduction in the cell, and it cause more glucose high in the body, and that lead to this type 2 diabetes. So this is what I have just told you. So and when type 2 diabetes, almost like 90% of the cases of diabetes are type 2 diabetes. The other diabetes are much less. I'm not going to those diabetes now. And the main problem, why I told you that this is a slow clear or silent killer, is that it develops over a period of time. It takes a long time, but when it start developing, it actually cause damage to different organs, and that is called a secondary complication, and that is more problematic. And when you are actually a diabetic, and if you're really a high diabetic patient, then lot of drugs you have a problem, then all these complications come. It itself doesn't cause it, but then uh, it causes the secondary complications which lead to even death. In fact, today in my last talk, I will just very briefly show you. In the first two talk, I will talk about the different diabetic pathways inside the beta cell, how we address that through mathematical models and network analysis. Two different I will show you. But in the last talk, very briefly, I will show you one of the secondary complication, which is the diabetic retinopathy. It's damage the eye. Blood vessels in the eye. Then what kind of problems we can address through our mathematical models. So to start with, this is not exactly, I will say, the network analysis we did, but of course it's a large scale model we used here, and it's the model is made from network. So what we did here, this is called the genome scale metabolic model, GSMM here written. So the aim here was to find the metabolic characters in the pancreatic beta cells of a patient with type 2 diabetes. So what is the metabolic changes happens in a pancreatic beta cell of a diabetic patient, which is different from a normal patient? Because if you, so this idea is to try to understand the mechanism, the changes in the pathway, because that will actually help you to find out the possible targets, drug targets, or therapeutic. So, in doing that, what we have taken here, we have taken these samples of online. This is an online data of 10 non-diabetic patient and 10 diabetic patient, and we have this is the pancreatic beta cell they have done, and they have this RNA-seq data. So this is actually a gene expression data they have. So of all these patients, they have this gene expression, that what are in the pancreatic beta cell, that what are the changes happen at the gene level. They have that data with. I'm not going in details of the data, but then we use this genome scale metabolic model. So this is actually you can think of an ODE in the sense of a larger scale, where these variables are the metabolites, and these proteins and all, you can think of the parameters. So all these, actually this metabolic pathway is one of the pathway in the biology which is well defined, that how these metabolic reactions are happening, what are the biochemical reactions are there, what are the genes or proteins involved in their reactions. So people have designed this GSM model. First model was the Recon 1, which was published in 2007. So they're taken from the experimental data and they build this model. So we have used that model here. The latest one we have used, not we have used the Recon 2 here, but actually now the latest one is Recon 3D. So Recon 2D has 7,000 odd reactions. It has 2,000 metabolites and 2,000 genes, and it looks something. Oh, sorry. It looks something like the x dot equal to ax, which I told you. Only thing is that when we start analyzing these things, we take it that the metabolites are at the equilibrium point. So this differential equation becomes a system of algebraic equation. Now the problem here is, since the number of equations and the number of variables are different, so you have infinitely many solutions. So we used this linear programming problem. Well, normally it was used. In this particular problem, we have not used that. We have developed something new, but normally people have used linear programming problem, and that is the objective function, and then they solve it. In our problem, what we have did, we use that gene expression data of 10 control and 10 disease. Then we use an efflux method to build the problem, build a model. And then we actually use this GSM model to calculate the flux changes. So flux changes kind of the reaction changes happening. And then we found that what are the reaction changes happening in diabetic patients, which is different from the normal patients. So we found that altered metabolic reactions. These are the efflux method we use. So interesting thing, what I want to show here, that when we get that gene expression data, this is the models which actually help us to predict them at the metabolic level, which the biologist can't get it from there. So this is, you can see the importance of mathematical models or the computational models which is coming into play in this kind of biological fields. So they came up to us with a set of gene expressions, and now there are so many gene expressions in a cell and so many metabolites or protein changes. But now with all these mathematical models, you can go back to them and tell them, for example, here what we have showed, that your fatty acid oxidation is very high, in case of the patient, the diabetic patient, then in case of the normal patients. Then you can actually tell them about what kind of reactions are changing in that one. I'm not going in details of the result, but the information which you can actually start from a gene expression, you can give it to the biologist. And the interesting part also you will find from here, for example, that model talks about that what kind of pathways which secrete metabolites or intake metabolites, for example, getting increased. So if we focus on the metabolites which is secreted from beta cells with high flux, I mean, in case of diabetic patient, these are the metabolites that get increased. This is one of the prediction which can made from this model. When we look at it, we get a very good result, in the sense that, say the second point, that we actually identify 7 secreted metabolites which get increased in the diabetic patient, which is known in the literature that they are associated with the cardiovascular disease. So this kind of information now you can pass. So you start from a gene expression with all these things. Now you make prediction that how, because when actually our body works, it's not that one organ works in silos. They actually, there is a problem started with one organ, but then start spreading to the other organs. And as I told you, as a secondary complication to the diabetes, cardiovascular disease is a major one. So this could probably give a link to them that, okay, these are the 7 metabolites which we observed to get increased in, or predicted, not observed, we predicted to get increased in the diabetic patient, can cause this disease. So we have published this one, but all the details, in more details is given there. Now this is an example of a computational model, for example, from a network, but we can make small scale models also, which, I mean, you have already seen in the ecology and all other places. So this small scale model which we can use here, I'm not going in details of this. Already have explained to you that how this type 2 diabetes occurs. And so here, one of the important part is that first you start with the insulin resistance that happens in different cells. Then it leads to impaired beta cell function. It actually reduces the beta cell function in terms of insulin secretion. And then, of course, it leads to type 2 diabetes. So one of the aim of this work is to understand this importance of impaired beta cell function, and to get the through the dynamics of glucose insulin secretion. So glucose is coming in the beta cell, and how different players are working in that one, leading to the secretion of the insulin from beta cell. So this is typically the mathematical model. Still it is very complex. As I told you, here we have to take that challenge, that how much complex we want to make it more realistic. At the same time, we can make it solvable also. But here, since the system is so complex, and we want to make it more realistic, so we take these terms, I mean, almost most of the major terms from literature. So in different places, these terms are given, where they have actually estimated these terms using experimental data. So we use them. So here we have taken 6 variables. plasma glucose to actually this will be the your readouts. Once you make the mathematical models, you must keep in mind that there are two important things are there. One is that what you are going to address, what is your question, so that will be your variable. So if you want to address that, I want to know how the glucose level increases in my blood, or I want to know what happens to the insulin level in my blood, so those will be, or what is the calcium concentration in this condition, so those will be your variables. And the other portion which actually connect these dots, these what the important players who govern these variables will be your parameters. So for example, when you say that you want to know the plasma glucose in the blood, then you need to know what are the things that govern this plasma glucose in the blood. Then you put them, for example, this first equation, you put all these parameters in terms of that, and then these are the relations with if that particular parameters has a relation with any other variables, then also you put them, and this is how you build up this mathematical model. So change in the glucose, plasma glucose, is governed by these four terms, for example. So and this is the schematic diagram of this model, so which I put. For example, here the glucose, which you can see here, one term, I'm just giving an example to show it. So increase in glucose, the change in glucose dgdt is now, if you see here, the insulin is actually inhibiting this. So this will be the term which we are using here at the last one, V14GI. So anyway, after developing this model, of course, there are two parts of the model. One is the differential equations which you build up. The other one is the set of parameter values, and this set has to be also, if possible, if you get it from literature, that's very good. For the normal patient or normal function or in any other condition, these parameter values, because each of these parameter value has a biological representation. If you see in the definition part, each of these parameters talks about some biological has a biological definition. So if you get those values from literature, you can directly put it there. Otherwise, if and of course you will not get all if you are making a new models and all, or if you're combining different things, maybe 50 or 60% you will get. The other you have to estimate. Now estimate is what? Now that estimation also has to be in a way that if you put those parameter values in your model, it must satisfy some real data. So we have taken that data from Dalaman paper, and they have actually give glucose and insulin, and we put the other parameters in such a way that they satisfied their data. So the red one is our estimated point, and of course here this is not I will not say this is the unique one, or statistically we have proved that this is the best one, but the idea here is to get at least one set of parameter values that fall within that range, because if we once get that set, that will be our best set to start with the simulation. And in simulation we will explore all possible parameter space, the whole parameter space, so that that will take care of this estimation, which I just told to you that this red light may not be your the unique one. So anyway, so you can see here. So after this, what we get here, that with our model equations and with our model parameters, at least the parameter set which have got, this is satisfying the available data. So once our model is validated, we start simulation. So we did a lot of simulations. I will also share the paper here. But here I'm just showing you one of them. For example, what kind of result you can get from there. So here what we have seen, try to take here is the loss of beta cell mass. So what will be the impact of beta cell mass? So in our model, V62 actually shows the beta cell mass. So what we did here, we showed here that if we change the beta cell mass to when it lost its function, so loss in beta cell mass, so when it lost twofold, threefold, fourfold, fivefold, we see that actually plasma glucose levels start increasing, because within each loss there is a decrease in the insulin level. I mean, this is expected result, but our model is also showing this kind of result. So once we get this kind of result, that yes, when we simulate our model, we get results close to observed result, then we actually can go for more details analysis and do something like this, two-dimensional parameter space, where actually we can see that, okay, here we took the insulin dependent glucose utilization one, I mean the rate at which the glucose is going inside the cell. We measure that with different other parameters to know uh, that what kind of effect of these parameters are on that one. So this is a two-dimensional parameter space we have taken, and the parameter selection I have not shown here as done using the sensitivity analysis. So you can use, we have used PRCC method to find the sensitive parameters. So we use that. So what is the goal of this kind of exercise? That once your model is validated, your model starts showing predictions. Now you actually start to identify the parameters which could lead to this kind of prediction, which could be more important in these kind of situations, because those hypotheses or those results will give you new biological result that you can actually share with your biologist. For example, here we actually focus on this red portion area, because this is the area which actually mimic your diabetic conditions. So we try to know that what could be the reason where we can reach to the diabetic condition, and then we did an exercise, I will not show here, that how we can bring the system back from that rate if it is possible. So in the next table, if you see here, I just summarize the whole result which we did. So we found that the reasons behind reduced insulin level and your, of course, increase in glucose, these are the reasons which we found here, if you see. And then through our recalibration exercise, we found that what could be the possible way we can restore it back. So we found that there are three parameters which biological meaning are like increase in ATP dependent calcium entry, down regulation in calcium excretion, and so on, extrusion, sorry. So those changes in those can actually bring back restores the some of the these observed so, restore back the diabetic condition in some of these observed condition. So we put it here, and just to tell you that, for example, we propose three possible strategies, but the first one, for example, is there are already drugs available which follows this strategy, where we actually ATP dependent calcium influx, they make the mechanism, they actually increase that, and they use this one. So for example, the other tool has not been used, and you can actually share with the biologist, and they can, and this can easily be done in vitro, I mean the cells, and then they can took in the animals. So as the last example, which I will show to you here, actually the main idea of this last work was to show you that whatever we have actually found in this work, we actually also validated with the biologists. The other works actually going on, but this validation is already published with the work. So what we here, the aim of this work is, as I told in the beginning, that we are looking for the secondary complication. Okay, there are reasons for the diabetes, and okay, diabetes is happening, but diabetes doesn't do anything alone. It has a secondary complication, and one of this is the diabetic retinopathy. So what we did it here, this is actually a typically network analysis, or sometimes known in the field of biology as systems biology, where we actually made our knowledge based network. Then this is maybe the next slide will be make it much. So we build a network using disease associated gene network. This is, so there is a database, one for the disease, one for the autophagy. Autophagy is a process inside the cell which actually help to clear your debris inside the cell, and sometimes also the foreign particles. So one of the idea of this work was that whether we can use this autophagy to have some kind of control in the diabetic retinopathy. So we use this to database to build our own protein-protein interaction network. We use different analysis. I will not go in details of this analysis, but one can think clustering analysis will be the topology, the hub nodes, and you have closeness, betweenness, distance you find of the network, all those things. So we did it. Then actually we use data, online data on DR. Again, there are 7 or 8 patients with diabetes, and the other 7 or 8 patients with diabetes with DR, the other one without DR. And then we analyze them and use the above protein interaction network to find the final target. So just briefly going in details of what we have done. So the first thing is that when we build that network without that, only using the disease database and the autophagy database, we found that there are 11 autophagic protein which are not reported in DR and are most central to the network. They are the, like, in the betweenness, closeness, what we have identified, they are to be very important proteins. So we took them as candidate autophagic protein, that okay, let's see what these kind of protein actually play a role in real data. Then we actually take a data from here. So these are the keywords which we search. We got this data GSE 146615, which as I told you, there are 8 individuals with diabetes with retinopathy and 7 individuals without retinopathy. And when we solved them, we went for the co-expression analysis and all, and then we also used a differentially gene express network. So in the co-expression analysis, we look for those genes which have same kind of expressions, and we do the analysis. Then we use here, we identify those genes which are showing higher in one condition, here the diabetic with retinopathy, then the other condition, the diabetic without retinopathy. And when we did all these analysis, we found that the CAPs which we have found there, out of them, there are there are 2 CAPs that found to be important. And out of that, we did one more analysis, random work analysis, where we actually revealed that T53 is one of the most likely protein that related to this one. So here I'm not going in details of the analysis, but you can see the, if you see the flow chart or written there, those are the things, once you make your network, those analysis you can do and actually identify an important protein which you can give to a biologist. And here we have actually did it experimentally also. So with the help of the biologist here in THSTI, we actually shows that if we inhibit T53, this is the inhibitor we have used, PFT-mu, it's a specific inhibitor to T53. It actually shows that it reduces the formation of angiogenesis. Angiogenesis is these are the angiogenesis you can see, the layer of blood vessels which form there. So if we inhibit that, it reduces that layer of blood vessels. So this is the experiment which we did here. So more or less what I try to show you through here, that whatever the mathematical methods you are learning here, it may seems very theoretical and all, but even say graph theory, differential equation, linear algebra, but they really have an real field applications, which is a need for today's clinical or medical field. COVID is a good example of that. A lot of theoretical results have actually was used when they actually go for this vaccine and all. So if people, if some of you want to take up this field, this could be actually a growing field, and when you actually take up this research, will be much more required in next 10-20 years. So before I finish this one, I thank my lab. So whatever I presented here, all the credit goes to my students and my collaborators, because what they did it, I just presenting on behalf of them. And this is the place we enjoy. If any of you are interested, you are welcome to visit my lab. And thank you for listening. If you have any question.
Thank you, Professor. Thank you. So this forum is open for questions, clarifications, and doubts. Sorry, my camera is not working. So
no issue. Actually, when I'm in the full screen, I couldn't see anybody.
Yeah,
I can see people.
You can see only the screen.
Yes.
So students, it's up to you. It's open for clarifications.
Yeah. No.
Or if you have some questions, you can also put it in the chat box.
They can also email me whenever they are. It's not.
Yes, I think they will.
Yeah.
So I have only one question.
Sure.
So this diabetics to mathematical equations. When you
somebody has put something in the chat box.
Okay. Let us see.
Yes. I don't know what.
Yes, one student has raised.
Okay.
Are you able to read the down?
Okay, I think he has no. She has written, sir, using the mathematical model, can we find the dynamics of as many numbers of variables we want at a time, or is there any limitation? No, sure. You can actually go for any number of variables you want. But as I told you, the challenge is that, for example, 2 dimension, 3 dimension is fine, but even if you go beyond 3 dimension, it's become difficult to handle analytically. I mean, you really solve it in pen paper. So you have to take help of the computer. Now in computer, the problem with, if you, for example, give an example of GSM. So here the variable was like in 2000, but the problem is that you will not get is as transparent as you get in the three-dimensional model, where you really have to see the variables, how it relates with the parameters. So you can see, for example, x star is equal to alpha plus beta, suppose. So you know alpha plus beta is controlling x star. In computational, you put a value of alpha, you put a value of beta, you can have you run it for 1,000 times, but still it's very difficult for you to get that kind of relation. But of course, if you really want to make some, it depends on the need. If you really want to go for a high dimension model, you need that to solve something, then you can go as many as variables.
Students, do you have more questions?
Anybody can please feel free to write me anytime. I will be more than happy to answer you.
How do you choose the initial data for the various problems?
Sir, actually, we choose the problem first and then look for the data, in the sense ki, suppose, for example, one of the problem which my lab is working on a diabetes. So we just look for the diabetes. We look, okay, we talk to the biologist. We understand, okay, this could be a possible potential problem. And there are two ways we get data. Sometimes, so we also get data from the collaborators or from inside. Otherwise, we then look for the online data if we get it. For example, the DR, there is only one data set has that data. So, and that was also very less, only 7 and 8 patients.
No, for the time evolution equations, you need initial data. So how do you choose this data? Where from you get?
You are talking about the initial time points,
Yes.
initial condition.
Yeah, because that's very important.
So initial condition, for example, this one, the model which I showed you. So we took it from the biological literature. What we thought, when, for example, after eating, after you have your food, so what could be the value of glucose that people have recorded? If we get from literature. Otherwise, the best part, if some biologists give us, because they have the experiment, they have their time points, so that would be more realistic, in the sense that they actually start from T0 and they give that this is the point we have taken.
No, but when you have non-linearity, very often they may be highly sensitive to initial conditions.
Sure, sir. Sure.
So you should have appropriate initial conditions.
Yes. So two ways. This is what you told about was very good, when the time point is finite, in the sense. But sometimes we take help of the steady state condition, and then that becomes independent of the initial condition. But otherwise, definitely, if it is like I'm looking for 8 hours, then that initial condition is very important. Otherwise, we get a different result.
Okay, thank you.
Thank you, sir.
So what are the number of equations you considered
Where, sir?
in your study? You have a set of equations in the
Yeah,
diabetes.
that is why I choose two different problems, just to show the students that it can be, for example, in the second problem, there were 6 set of equations.
6 set of equations.
But in the first, there were 2000.
Okay.
So the first one is more computational.
Mm.
And the second one is, we did something by pen paper also.
Okay. So there is one more question in the chat box.
Oh, okay. Does we get any other model other than genome scale model in biological modeling? Sure. So there are many more models are there. I mean, depends on what kind of. So genome scale metabolic model is one of that is available. You can build your own model, and there are other models are also, but mainly on the metabolic part, because protein is not that well defined. Because to have the model, you need to know the direction. So X is changing Y, that you need to know, which is not that well defined in case of protein. But yes, you can get. The last one is, sir, can we implement general network model in biological modeling? This I didn't get. Can somebody explain what implement general network model means?
Kausalya, can you unmute and ask the question more precisely? What do you mean by general network model, Kausalya?
Okay, I mean, if I can get it, I don't understand general network models, but you just think is that same as mathematical models. So the basic difference in network analysis, if I can tell the students in that sense, is that it will give you more static in nature, unlike the mathematical model, which is more dynamics. I mean, that is something changing over time. So other than that, I mean, you build your sorry.
He has written something more.
General network no, then also it becomes specific. Yes, yes, of course you can use it. It's actually depends on the data. It's mainly the biological data is very complex. I mean, it can be, if you are looking at the cell numbers, it's a different data. If you are protein numbers, it's a different data. But of course, people have used these models.
Is that clear, Kausalya? So, are there more questions? Maybe they will write to you by email.
Sure, yes. Actually, I have introduced a lot of things today that I later realized.
Sometimes students hesitate to ask.
No, sure.
Okay. So since there are no more questions, I would like to conclude the session by thanking Dr. Chatterjee for accepting our invitation and giving a very wonderful talk. It will be really useful. Sir, thank you very much.
No, I thank you, and of course, university for giving me this opportunity. I always loved to have an interaction with the students. Thank you for giving me the opportunity. Thank you.
I will write you the other details by email.
Sure. Well, and you can also share the number. I just forget to take.
Oh, sure.
I will do that. Thank you, sir. Thank you.
Bye.